We're now moving into a case study part of the afternoon. We're going to hear two presentations that are cases, and then we're going to hold the questions until the end of both presentations. But I'd encourage you to keep sending in your questions for the first presenter following her talk. Our first presenter is Dr. Dana Abramovitz. She's Senior Director, Scientific and Corporate Strategy at Strand Life Sciences. Strand is a bioinformatics company whose products include array, mass spec, and NGS analysis. They're used worldwide, and they've recently begun moving into the field of clinical diagnostics. Dr. Abramovitz leads Strand's U.S. growth into the clinical genomics and healthcare industry, and this case study is called Accelerating Disease Diagnosis Through Automated Genomic Analysis. Please welcome her to the stage.
Hi. All right, so first off, I want to thank the conference organizers. This has been a fantastic conference, and then also a great team of people to work with. So let's see, I guess I hit that button there. Okay, so I'm actually, first of all, I have to disclose that I am an employee of Strand, and I'm going to be presenting a case that is a collaboration between a clinic and a company. So before I get started, let me give you some insights into the players in this case. So Strand is a bioinformatics company. We have life scientists and computer scientists, and we kind of mesh them together, and that's what that image is representing. We recently moved into the clinical space, the clinical genomic space, and have labs in India. And it was through those labs that we started this collaboration with the Center for Human Genetics, Bangalore. And CHG is a research institute that's staffed by faculty from around India who are interested in genetic diseases. And we so one of the things with working with them is that a lot of their patients tend to be lower income, and so being able to do this diagnosis in India is really exciting. So one of the physicians that we worked with at the Center for Human Genetics is Dr. Banakshi Bhatt, and she is a practicing clinician at the Bangalore Fetal Medical Center. Okay, so I've been asked to prepare some learning objectives, and the first one is to be able to describe the genomic analysis pipeline, and I think that we've seen that recurring throughout the talks. It was certainly present in the plenary sessions this morning, and then I think the other objective is the ability to recognize how genomic interpretation software, and certainly the importance of the bioinformatics to help with the data analysis. And so hopefully we'll get all of that covered in this presentation, but I do believe that Dr. Jacob presented it excellent this morning. Okay, so this particular case, we're looking at a young family a young couple that's trying to become a larger family. And so they're healthy parents, but they're in a consanguineous marriage. And this is fairly common still, either from culture or religion or even geographic location. And they've had 2 children. Both are boys, and they died before they reached the age of 2. And so they're trying to grow their family, and Dr. Bhatt had been working with them throughout 4 or 5 years, and they approached her to try to use genomics as a way to help guide future pregnancies. So this is the clinical presentation of the children, and typically when you show the clinical presentation, you black out the eyes. In this case, the eyes are the prominent feature of the phenotype. So with these boys, they have exophthalmos, which is the bulging of their eyes. They both had respiratory distress issues, a depressed nasal bridge. And Dr. Bott spends a lot of time looking at the patients to really evaluate their clinical presentation. And she can tell a lot about them just by looking at their faces. However, just really understanding the underlying issues that led to death and how we can use that information to guide future pregnancies, we looked at the genomics. And so when the second son passed away, Dr. Bott had the insight to take a DNA sample. And so we actually did whole exome sequencing of the trio. And I know that there's a lot of discussion, when you're looking at a rare disease diagnosis, do you use whole exome sequencing? Do you use whole genome sequencing? In our case in particular, because we're trying to keep costs to a minimum, because of the situation in the family, we really wanted to, we chose to do whole exome sequencing. This also been shown to be widely used and, sorry. Okay, so this is our genomic analysis pipeline. And so when you do your next generation sequencing, you get these different reads, and you have to initially do your alignment to put together your genome. And then, and so depending on the type of instruments you use, you might have different protocols for that. But it's pretty standard, and the instrument pretty much tells you what to do. Then there's variant calling. So to align your aligned genome against a reference, and then to identify the different variants. And so there are a lot of commercial and open source mechanisms to do that. We used one. There are lots of them out there. So the next things that we did were really in collaboration with Dr. Bott. So going back and forth and trying to hone what was acceptable in order to try to find that culprit gene and that culprit variant. And so first we filtered out common variants, and so we do that by comparing to 1,000 genomes, dbSNP. We actually have our own internal ethnic genome reference based on the multiple cases that we've done already to filter out the common variants. And then we identify the variants that cause a protein change. So these are the missense mutations or the splice site variations that could lead to a truncated protein or a protein that may not fold properly. So starting to identify the biological implications. Then we prioritize by the family pedigree. So in this case, we do have the information because we had the family. We knew that the parents were healthy. We had 2 sons that both died, and so we were able to kind of determine could it possibly be X-linked? Is it a complex heterozygous mutation that we're looking for? We can kind of plug that in. And so that's all stuff that a lot of the bioinformatics teams typically do. And so as clinicians, when Dr. Bott, she has this phenotype and symptom information, wants to incorporate that as well. And so that's the final and in some ways, the most important step is including the patient's presentation. Okay, so when we run our pipeline, we were actually able to narrow it down into 6 candidate genes. So we're, through this collaboration, we're learning through this experience and putting it into software. And so this is the representation of our candidate genes in the software instead of an Excel file. And so you can see that, let's see, there we go. Oops. So there's the patient phenotype information, but then there are the genes, the candidate genes, the different diseases, the different clinical manifestations, so like the organ systems that the genes are shown involved in, and then the disease variant density, so how many variants have been identified in a particular gene that have been found in the literature to be associated with disease. And then these darker lines, and I don't know if they're visible on the screen, are where our particular mutations are located. Okay, so then we can look at these more carefully. And so the first gene, FLNA, we look at the diseases, we look at the clinical manifestations, and they actually match some of the things that we've seen in the children. When we look at the next gene, the disease is not a relevant phenotype. The next gene, also not a relevant phenotype. We have a gene that's not in a relevant tissue. And then finally, a gene that there's only vague reports of the phenotype. So we tend to rule these out. Then we looked at the last one, FLNB5, and that again has some phenotype information and also clinical manifestations. So we decided to focus on these two genes. Now, this view here is just showing the filtering at the gene level and not actually looking down at the variants. So we need to do a closer inspection of that. And so that's what we're doing here. And I should mention that this is still in progress, but you can see that this particular variant causes start loss mutation, which was considered not damaging by the predictors. But when we look at, and this is now a display of the protein, this guy here is the mutation that's in our case. So it's a methionine to a valine mutation, which would prevent the protein from being transcribed. However, our algorithms identify that there was a second start at this position here. So it is possible that the protein was actually made. Now then the question is, is that resulting protein functional? And so we can look at different variants, and so that's what so here are there are two diseases associated with this gene, macular degeneration and cutis laxus. And then we can look at the mutations in the literature to see if these mutations have, so what's been found for these different mutations, and do they match up with the phenotypes that we've seen in our patient? So they didn't. In most of the cases, all the results showed cutis laxa, which is a elongation of the skin, and we didn't see that. That's not a phenotype in our patients. And then the concern was, well, is there a problem with the first integrin binding domain? And so we actually looked at a mouse model to show that if that integrin binding domain is missing, then you get cutis laxa, which is, again, not a phenotype that we see. So we effectively ruled out that particular protein. So now we look at FLNA variants. And so this one, it's a missense mutation that changes an aspartic acid to asparagine, and it's predicted to be damaging by 3 of the 5 predictors. So this is a gene that has many more variants associated with it, lots of diseases associated with it, as well as a range of binding partners, and so that's what this information is here. So the mutation in our case is this aspartic acid to asparagine here. Let's see. And then we can look and see what is actually happening, and could this gene be relevant in this case? And so we have our phenotypes, and we can see, has this gene been implicated in phenotypes that have been seen before? So, for example, has this gene been involved in pulmonary atrial hypertension? And it has. Has it been involved with a bulging of the eyes? It has. What about early death? And that's been observed as well. And then we know that the mother is heterozygous for this gene, and carrier females are subclinical. Let's see, go back. So when we look at our particular mutation, however, we see that it's pretty much isolated here in this location. So all of the known variants, or at least variants that have been reported in the literature, are on either side of our variant. And so we wanted to look at this because it hadn't been seen before. And so we look at the conservation, and we know from the function, so aspartic acid's acidic, asparagine's neutral. This could affect the structure and function of the protein. It's conserved, it's highly conserved across all species, which suggests that this is a pretty critical mutation. So I know Dr. Jacob mentioned earlier this morning when they were looking at a gene that they had, they didn't have much information for it, but were able to look to see, other binding partners. So we usually do that as well. And so here are the binding partners where the particular domain that's mutated in our protein is interacting with these particular proteins. So we're able to look at the other proteins that are binding partners to see if they are producing similar phenotypes if that binding interaction is not able to take place. And so we did see that that was the case. And so we felt pretty confident that this was the particular gene and the particular mutation that we are seeing in the patient that's leading to the pulmonary problems, the bulging eyes, the facial distortions, and the early death. Let's see. So I said, we've done the trio, and the mom is heterozygous for this particular mutation, and FLNA being on the X chromosome pretty much sealed the deal for us. So we felt really good that we had identified the gene. So now incorporating this into the decision process. So as we're working on this, the couple conceived and came to Dr. Bott and wanted to know what should they do. We had just identified FLNA as the likely culprit, but we knew that FLNA also showed some skeletal manifestations. We didn't have radiology for the deceased children. The mother was pregnant, so we weren't able to do that on her, so it was kind of a concern. However, we did do ultrasound on the fetus at 12 weeks, and it showed some abnormalities that could be considered associated with Down syndrome. So that was enough to warrant doing DNA testing of the fetus. And so we did do that. And we found that the fetus was heterozygous for both FNLV5 and FLNA mutations and negative for Down syndrome. And in fact, the genotype had the same exact genotype as the unaffected mother. And I'm pointing this out. So in India, you're not allowed to report the gender of the child. And so Dr. Bott saw that there were 2 X chromosomes, and the fetus was heterozygous. And after a second ultrasound at 16 weeks, it showed that the child was developing normally. So those initial abnormalities had gone away, and recommended to the parents that they continue with the pregnancy. Now I do have a happy update. A baby girl was born August 26th. Unfortunately, I don't have any pictures of her, but the mom says that she doesn't have the same appearance as her brothers. So we feel really excited about that. So just to summarize, so we used whole exome sequencing to focus of the trio to really narrow down. We used our genomic analysis pipeline to get a list of candidate variants. We then incorporated those steps. So going back and forth with Dr. Bott, we were able to incorporate those steps into software to kind of make that information easier, and all the literature searching that has to take place is now consolidated in one place. And then we informed the patient of the results and followed up with imaging. So now we're trying to make this information more accessible to clinicians. So, it was wonderful to be able to collaborate with the Center for Human Genomics Bangalore, because it really helped educate the software. But it was also good for them to interact with the company, because it helped explain, show them what was possible. And like what could happen if you have a team of bioinformaticians that you normally don't have access to that could really help you. And so we've combined that into software, and so that people who don't have teams of bioinformaticians could actually do a lot of this work. And we're introducing an early access program this fall. And so, it'd be great if any of you are thinking about doing this type of thing. And I should point out that although the case that I talked about was for the diagnostic odyssey in rare diseases, we've actually used this pipeline for other diseases, so cardiomyopathies, autism, various ocular disease. So it's possible to use that. So if you have any questions, you can email me or I'll be around. Thank you.
Thank you very much. Dr. Abramovitz will be out in about 20 minutes, and we'll take questions together. Our next case is going to be presented by Dr. Douglas Riegert-Johnson. He's consultant of medicine and medical genetics at Mayo Clinic in Florida. After medical school at UAB, he trained in medicine and gastroenterology at the Mayo Clinic here, and in medical genetics at Johns Hopkins. The medical genetics group at Mayo Clinic Florida is focused on cancer syndromes, and his case is called Precision Diagnosis of Glomerulopathy to Prevent Occurrence in Subsequent Generations.
Good afternoon. It's my pleasure to present a case from Mayo Clinic Florida to you. First off, I'll have to apologize about the title. I tried to think of a catchier title. My wife said if she saw that title in a program, she would turn around and go the other way. And she's not here today. Two learning objectives: to know some of the characteristics of whole exome sequencing, which have been also reviewed in other talks here today. And I'll also say know some of the characteristics of pre-implantation genetic diagnosis. I think it's important for everyone who's involved in genetics to have some appreciation of PGD technique. With that being said, I'll give the disclaimer that I'm not certified in maternal fetal medicine and I don't perform PGD. Our case started with a 28-year-old man who presented to the clinic with a large amount of protein in the urine. And based on this, he was diagnosed with a disorder of the glomeruli of the kidney. Those are the small units of the kidney that perform filtration. And I will show you a picture of the patient's glomeruli. First off, I'll show you a picture of a normal glomerulus, if I can get the cursor here. You can see that there are these wide open, we call lacy open capillary loops where the blood flows through, and then different electrolytes and other things dilute outward into the capillary urine space. And here is a picture from the patient's own kidney biopsy. And you can see these wide open lacy loops have been replaced by this pink staining material in a lobular format. So it wouldn't be, you wouldn't be surprised this patient would have protein in their urine. Going in for a closer view with electron microscopy, there are some finely granular deposits. And the endothelial cell, which is the cell lining the wall of the blood vessel in the glomerulus, are here. And the basement membrane, which is an integral part of the filtration, is here. And in between the two, you can see this area I've shaded yellow. And those are the granular fibrillary deposits, which are resulting in his proteinuria and kidney dysfunction. So now we have identified that the patient has subendothelial deposits in his kidney that are most probably the cause of his kidney dysfunction. How are we going to identify what that compound is or protein is? Well, we use another ohm. We won't use genomics. We use proteomics. And first off, microscope slides will be made from that kidney biopsy. In small areas denoted here will be microdissected out, and using tandem mass spectrometry, the proteins in those microdissected out areas will be determined. And you can see the readout of that here. The first protein noted is fibronectin. And if we go over to the relative signal strengths of the different proteins seen, you can see that fibronectin is by far the most common protein in the sample submitted, which is and is present in much greater amount than normally seen. So now we have a patient who has glomerulopathy with fibronectin deposits, which is a known disorder, which goes by several acronyms, one being GFND. And GFND is a rare disorder, but it has been shown to be associated with mutations in the FN1 gene, which you would surmise correctly so codes for the fibronectin protein. Enter the picture a spouse. The patient's married, and the patient and his wife wish to have children. And they wish to have children who have normal DNA. And this is my daughter, who I would say has normal DNA, playing with her DNA floaty in the pool. In our house, we have all kinds of DNA-themed toys, you know. The problem with regards to that is that GFND is a dominant disorder. And so any offspring from our unfortunately affected patient would have a 1 in 2 or 50% chance of inheriting GFND. And he and his wife would like to change those odds in their favor. The solution to this problem is pre-implantation genetic diagnosis in many cases. And in order to proceed with PGD, a precise diagnosis is needed as a prerequisite. So there probably are many people in the room who are familiar with PGD, probably many more familiar with it than I am, but I will review this. So PGD is a technique which was first described in the late 1980s. There have been over 10,000 pregnancies assisted with PGD technology, and the cost is approximately $3,000 to $7,000 in addition to the cost for a typical IVF cycle. And to walk you through the steps, first an egg donor is given fertility drugs, just like in IVF. Multiple eggs are produced. Eggs are fertilized as embryos. Then some of those eggs are tested. And in this case, it probably would be on day 3 in the morning. The 8-cell blastomeres would undergo a single-cell biopsy. And that single cell would contain a tiny amount of DNA, 7 picograms, that would be tested for the genetic mutation that we're interested in. And then only the embryos which test negative for GFND, or the genetic disease you're interested in, would be implanted, and then the mother would give birth to a genetically healthy baby. Now, I will say that this is a little bit misleading, and the patients are often under the impression that if they go through this whole process, the child will be genetically healthy. But you're really only screening for GFND. So the child is unfortunately still at risk for all the other disorders which affect children and adults. So unfortunately, the 1 in 50 or to 100 risk of autism, other risks for malformations, classical genetic diseases such as PKU, hereditary cancer syndromes, and so on. So here's a picture of a blastomere biopsy. You can see that the blastomere at this stage contains approximately 8 cells, and a small tunnel is drilled in the zona pellucida around the egg, and a single cell is removed for diagnosis. So I'll come back to the family tree, and as I said, GFND is a dominant disorder. So here's our patient. His wife's unaffected. He has an unaffected brother, unaffected mother, but his father had a kidney transplant at 52 for a kidney disease which was never clearly diagnosed. So at this point, we would assume it was also, GFND. And then we have the 2 proposed offspring here. So what are the challenges? We need the precision or precise diagnosis of the exact FN1 mutation or GFND mutation causing disease in this family. Well, there are 2 challenges. One is that at the time this patient was seen, there was no laboratory in the world that was offering testing for FN1 or other genes associated with GFND. And even if available, studies in GFND families found only 4 in 10 had a mutation in FN1. So there are most probably other genetic loci in play, i.e. genetic heterogeneity. So the solution in this case was whole exome sequencing, because it will test both FN1 and it will test all the other genes in the genome, or let's say 95% of the other genes in the genome as well. So samples were taken from multiple family members. You can see them denoted here with the asterisk, the affected patient, his affected father, and his unaffected brother. And technical note, whole exome sequencing was performed on all 3 of these individuals simultaneously. No arrays were performed. There was no targeted sequencing done at a later date. And here's the report from the laboratory that performed the testing. And I will go through some of the things that are reported in this report for you. You can see it's noted as positive with relevant alterations detected. It's late in the afternoon. Does anybody know who this is? Anybody have young children? This is Disney Princess Belle, and she looks like she's frightened. What is she frightened of? She's frightened of this 7-page report that has writing so tiny that when it's faxed, you can't read it. Or is she frightened of my son playing the Beast at Disney? You can decide. So we'll just walk you through just a few characteristics of this report or factors about this report. First off, the cost. So when this test was ordered, it cost $10,000 cash. And I've been told if we ordered it today, it would be $15,000 cash. Insurance did not cover the cost of the testing. Turnaround time, 6 months and 13 days. And then, of course, we have the positive test result. So the people who wrote that report did a very good job of going through the rationale why this mutation that was identified, which is tryptophan at 1017 to cysteine, was the disease-causing mutation. And you've heard today multiple discussions of how that is done. In brief, the criteria, which I've developed my own acronym for, is that the mutation's been reported in a characterized gene, so that's more likely it's disease-causing. There's a class change in the amino acid, and the amino acids conserved. You can see this Venn diagram here shows the different classes of amino acids, and tryptophan is here, and then there's a cysteine with a disulfide bridge and a cysteine by itself. They're both in different classes, and the gene's been associated with the phenotype, and FN1 mutations have been associated with the phenotype. In silico testing was positive, and this mutation was not present in any known database. And I would say most importantly for a mutation that's been reported before is you can demonstrate segregation, which we were able to do. Now this patient was very satisfied, and his wife was very satisfied with the outcome of this testing, and they agreed to make a nice video for us that showed their feelings about the testing and how things had gone. And I will ask the audiovisual staff to roll the film.
I don't think my outlook on life has changed really at all. I can pretty much see the silver lining in anything. We knew that the disease I have is slowly damaging my kidneys. We just didn't really know the cause of it. The care I received before coming to the Mayo Clinic, there seemed to be very little proactive investigating as to, let's be more specific, let's go a little bit further to figure out what this might be.
When we got married, I knew he had kidney disease, but neither of us expected it to be as serious as it was so early.
There's potential for me having a transplant in 5 years, 10 years, 6 months, we don't know.
When Dennis came to Mayo Clinic, the most important thing was to make sure future generations didn't have to face the same task of potentially having to undergo renal transplant.
My wife and I have talked about the potential of being able to pass this on is it right to do that knowing there's a possibility for, our children to have this disease?
The question was just what exactly is causing the kidney disease.
They said, look, we want to run some tests, we want to do another biopsy, we want to figure out what this is. And I said, great. My wife and I are totally on board.
We actually went with something called whole exome sequencing, which took the participation of him, his father, and his brother to actually identify the FN1 gene mutation within the family.
They found that gene, and it turns out, sure enough, it was mutated.
Now that we've gotten the test results back and we know the gene that is affecting him, we can make an informed decision about having a kid.
This positive test result gives him and his wife the option to pursue in vitro fertilization followed by pre-implantation genetic diagnosis to try to ensure that the next generation actually doesn't harbor this FN1 mutation themselves.
The care and the information I've received from the Mayo Clinic has been really fantastic. To have the confidence in the people who look after your care is very settling.
Mayo Clinic is always on the forefront, pushing ahead, trying to determine how the newest and greatest technologies can benefit not only us, but more importantly, our patients.
As you can see, the patients were very satisfied with that outcome, and I wish that was the outcome for all of our whole exome testing cases. And with that, I'd like to acknowledge the entire SIM team, with special thanks to Megan Roberts, who's the genetic counselor you saw in that video, and Princess Momo and Princess Audrey wish you a nice stay in Rochester. Thank you.
All right, well, we have questions on both cases, and continue to send those in. The audience has stayed with us here for the last presentation, so there's a whole bunch of questions. One thing that struck me though when I was watching both of your presentations is that the amount of resources and expense that it took for each of you to accomplish something pretty miraculous. Who bears that cost? And going forward, I mean, is there a model to absorb that?
So
either one of you can comment first.
Yeah, no, I was going to say, I think that the families are the ones who, have the most cost, and that's what we're trying to, kind of reduce that for them. In our case, it was. So it was through that collaboration that we had, we did the sequencing and then the software development, and then the idea to use the lessons learned into a software to reduce that cost.
Uh-huh.
Well, I want to second what you said is that we should never forget that the true cost of this is the cost of disease which the patients are bearing and their families. In terms of the logistical cost for us, the cost of testing the patients bear, their insurance companies bear, and we've had some success with that. The personnel cost, it's still unclear. I would honestly say completing all of the work needed to do that for that family, again being one of our first whole exome cases, probably was a full 40-hour week for our genetic counselor. And obviously the family was not billed for that, but we're still struggling with how to gain efficiency without compromising care on many different levels.
One of the, there's a whole series of questions, but one regarding your case. Where was the patient sequenced? Was it in India or the US? And how did the information transfer, both in terms of data and ethics? How did that work?
Right. So all the sequencing, like the whole case was actually done in India. So our lab there, and then my company's headquartered there, and so we didn't have a transfer issue because our lab is actually like in the same complex as the clinic. So that was pretty easy. What was the next part of the question?
The ethics, were there, in terms of informed consent and those kinds of issues, were there? differences in having to work internationally?
Right. Yeah. So we definitely have. So it's a completely different situation in India. The regulations are different, but we are a global company, and so we uphold to the highest standards, and so we're looking at the regulatory guidelines in the U.S. And Europe, and so using those highest standards.
Question for you: how can a blastocyst successfully generate a healthy fetus after loss of 1 of 8 cells?
Great question. I wish I knew the answer. I only know that it's possible, and PGD biopsies are not they are associated with increasing loss of the embryos, but they're not associated with any sort of increased risk for fetal malformations or other health problems outside of what would be seen normally with assisted reproductive technologies. Now, this conversation would be outside the scope here, but there's thought that any type of assisted reproductive technology, IVF without PGD, which is used very commonly, might place people at increased risk for certain disorders of methylation in complex genetics like diabetes and heart disease later in life.
Do you know, is this the first time whole exon sequencing was done as part of PGD, or is that something that's done?
That's a great question. Yeah. I don't know the answer to that. Now this was done over a year ago, so it may have been one of the first cases. Yeah.
Typically, it's targeted testing for a known gene.
Mm-hmm.
Yeah. It's a lot to read through here. Okay. Someone asks, should I get DNA samples from my living parents just in case I might need it later?
That's a good question.
Should we be storing DNA from our parents or siblings?
I've often thought about storing, DNA from other family members. I haven't crossed the activation energy for that, even though I know how to do it and will go forward with that.
Yeah.
Could go forward with that.
Yeah, no, I think it goes back to a lot of the discussions earlier about, patients and patient consent. I know that, so my mom is a breast cancer survivor, and, this is before, when the BRCA testing was just getting started. Mm-hmm. And we worked with a genetic counselor, and she was offered the testing, but she just didn't want to know. It was it was scary for her, and she didn't want to know. So I wouldn't I would have to have a conversation with my mom, with my parents, to see if that would be something. Yeah.
How do you account for the great genetic heterogeneity found within India? When removing common variants?
So can you repeat that?
Yeah. Yeah, that's right.
Yeah. So we have the databases of information that you can subtract, right? But the entire Indian population is not included in that, and so it's difficult. And what we're doing is doing the testing and storing that information in a database that we can then use to help filter out. We're building our own and making it accessible, and that's one of the things that is really key. That came out of our learning experience was the ability to, once you identify new variants, to be able to put that back in to help inform subsequent analyses. And that's my thinking is that the more sequencing that we do, the more that we learn, it's only going to get better.
Mm-hmm. Mm-hmm. question for you. Can sperm be tested for the kind of disorder you presented? Unaffected sperm should be 50% be used for in vitro fertilization, so to avoid having to perform blastomy or biopsy.
No. I think the sperm would be destroyed by the testing. The couples in this situation have several options, and I'll just go through them. One would be not to have children. Two would be to adopt children. The third would be to conceive children using sperm and eggs from other partners. So sperm donor or egg donor, either carry those, the female in the relationship could carry those pregnancies herself or through a surrogate. Despite offering those options to many couples, there is an innate desire to have your own children, I have found. And although all those options are reasonable and available, a minority of couples select for any one of them. There is a similar option raised by the question here, but not for sperm, but for the eggs. So there are entities called polar bodies, which are small, you could think adjoining fragments of the egg that are generated when it is made, and you can sample these. This would be polar body testing.
What's the advantage of that over what you did here?
Polar body testing advantage would mean that you don't actually have to sample the embryo. And when you sample the embryo, it's associated with increased loss of those embryos. So you can do that test before you generate the embryo.
This is for you, Dana. Is it possible to use cell sampling from early stage embryos in India to identify safe embryos to implant for couples like the one you described here?
I would imagine so. Yeah.
You know, is that a strategy that's been done for this?
I don't think so. I think it's just like the cost of in vitro fertilization and like the creation of the embryos. I just think that would
make it prohibitive.
Yeah.
In terms of cost and time, how long did the pipeline take for your project, and how are you working to minimize that?
Yeah. So through the collaboration, it took several months of going back and forth. But we've been able to automate the pipeline, so now you can actually run it, and it only takes 30 minutes. 30 minutes.
So if you were faced with a similar situation, how long would the whole process take?
So like from like what, 10 hours to do the exome sequencing. Using the alignment software that we have, I believe it takes like 4 hours to do the alignments, on a multi-core machine, and then the analysis. So, you could have a 1 or 2 day turnaround.
DNA banking is encouraged in patients who have a hereditary type of cancer and a family history, but have had negative genetic testing. What are your thoughts about that?
Depends on the case situation to speak. My first thought now, when a patient comes to me who has a hereditary cancer syndrome pedigree, but's had negative testing in the past, is should we now repeat the testing using newer technologies? Because it's very likely that some genes that are now available through perhaps next-gen panels weren't tested. It's also a thought that maybe tests for large deletions and other rearrangements weren't available when they were tested. So my first thought would be, what was their testing and was it complete? And then in terms of banking their DNA, we do offer that to patients. For reasons I haven't been able to put my finger on, I can't recall a patient who opted to have their DNA banked for hereditary cancer syndrome.
You haven't seen that?
I can't put my finger on that.
Well, someone asked about banking. If we do get DNA on relatives, how and where can it be stored, and how expensive is it? Is it something that there are clinics and laboratories that offer that service?
There are definitely laboratories that offer that service on a commercial open access basis, and they are available on the internet. I won't go mention any names. The Mayo Clinic's not one of them. And you contact them and they will send you a kit, and some of them use blood samples, some of them use saliva samples. The fee, from my recollection, is, considering the cost of the other things we're discussing, is nominal. It's several $100s for a period of time, and my recollection is the one lab we had recommended was 10 years or more. And then in terms of accessing the sample, that was who had permission to access your sample and how was included in the consent process that which was sent to you in the mail via the laboratory. So maybe the reason I haven't don't have a good feeling on the uptake of that is I've been had to refer patients to elsewhere, and I don't know if they follow through with it or in what way.
Dana, there's a lot of questions coming in about issues of collaboration cross country. This one, in consideration of the cultural differences you face collaborating with a laboratory in India and the high cost of the tests, do you see such types of studies or collaboration being extended to other developing nations anytime soon? I mean, were the barriers very challenging, or they're just things that you had to learn to work through?
So in our case, it wasn't challenging because we've been established in India for 10 years, so that part was fine. And like going to different countries, I think that it would be possible to do. Yeah.
Okay. Did the couple in the case study undergo PGD? I think you said they did, right?
They went to a PhD clinic, they were consented for the process, and now they're considering their options. So they haven't gone through with the testing yet. I haven't spoken to them specifically about that, but from other couples, a couple of things are probably going through their minds. One is the health of the patient. He has a serious medical condition, and whether they want to start a family at this point with him considering having a kidney transplant in upcoming years, and also the cost. One cycle of PGD with IVF with PGD would be $20,000, and the success rate for that for having a pregnancy with that is going to be less than 100%. So you could easily pay your $20,000 and then have to come back and pay it again and again and so on. It's less likely with this couple because they're not known to have a infertility problem.
One of the things that was raised this morning, Wiley Burke talked about in terms of ethical issues, is diffusion of technology, especially very expensive technology. I mean, what solutions are there to make promising technologies available to someone who can't spend $20,000 out of pocket to have a healthy child? Are there only options the other choices that you had of adoption or not having children?
Well, I think increasing competition has been shown to decrease costs, and there is in this country some transparency about the success rates of IVF clinics. So there are reports generated by the NIH, which are available to anyone to see, that will show the reported success rates at different clinics, so you can compare across that. But you're right. The costs are high. The median family income from the United States is $51,000, so $20,000. That is a lot of money. Not to mention raising the child afterward. So, as I speak from experience, the other options are often used by families. Many people, feels the pressure. Amount of pressure is not the right word. Many people opt for not having children in some of these situations, considering all the things that go into that.
Any comments on that? On the cost issues? I mean, you're developing new technologies. Over time, do you see costs coming down, or?
I hope so. I mean, in order to make, I'd love to see this available to everybody, everybody who wants it. So, I hope that, through technology, through software, through consolidation, through optimizing processes, that we can get costs down.
Well, I want to thank our two speakers for presenting their cases. I think over the course of the day, we've come from very big picture down to seeing how genomics and these technologies can impact on one family, on one child, on one life. And so it's been I think a nice arc to the day. So thank you both very much.